For coming this afternoon on a Friday, very chilly evening today. So thank you everyone, thank you to the students and the judges. But this is a tradition that we have been following, and many of you are from outside of UNC Charlotte. So first and foremost, I would like to welcome you all to UNC Charlotte, this beautiful campus. And this is our 9th annual 3MT program, which is called the 3 Minute Thesis competition. So as you know, the 3 Minute Thesis competition is a research communication competition in which graduate students compete to explain their research to lay audiences in just 3 minutes. So it's like a pitch that they give you, and using only one slide. So this is kind of a difficult thing to do, and it's wonderful that a lot of our students participated. We had 60 students participate this year, and out of this, all of them were just wonderful. And out of the 60 students, there are 10 who have come to the finalist, who have risen to the finalist level. So if there are any students that were there for the preliminary round, I would like to give you a round of applause. Thank you very much for participating. Now next, I want to acknowledge a very special person who has recognized this event, and his name is he's Raju Mahajan, and he's from Raju Law. And he has been sponsoring this event for the past 2 years, and we are really, really grateful. Unfortunately, he is not able to be here, but some of his colleagues are here who are sitting on the back over there. Thank you for coming. So their names are Chelsea Smith and Amanda Schism. And our relationship with Raju Law goes back to 2020, when Raju began facilitating immigration visa workshops for our students, sometimes accompanied by Chelsea as well as Amanda, who are here. It was during one of those workshops that Raju mentioned the value of being able to convey research in a manner that the general audience can understand. So as a follow-up, Aura, who's here, give applause to Aura. Hello, Aura. So Aura mentioned 3MT to Raju, and he jumped at this opportunity and said, I want to sponsor this event. And so he's been doing that since then. So since Chelsea has also facilitated this communication workshop, we are so grateful that our students today have a sponsor for this wonderful event. So we have nice food and everything after you finish. There are many other people to thank today who supported our students as they prepared for this competition. Doing something like this stretches from outside their comfort zone, and support and encouragement that they get is really, really important, from their mentors to their faculty who support them. But really, I would like to take a moment to thank Dr. Elise Demeter for facilitating a workshop again this year and preparing our students for 3MT. Thank you also to Dr. Derek Kerr, who made himself available to all 60 preliminary round participants for coaching and worked closely with these 10 participants, or these 10 finalists. Thank you also to John Howard, who will be in the audience. You could have him stand up. Thank you very much. He's the creative director for Crafted Volunteer to review and provide feedback on all of the slides that the finalists will use today. We appreciate everyone's help and support. Thank you to all the 20 or so people who served as judges in the preliminary rounds. Thank you. We also appreciate our judges today, partners from Charlotte community who are dedicating their time to be part of this. As you see, they are the perfect people to judge because in their professional roles, they use data and research to make important decisions every day. The first one, Glenn Kowalchuk, interventional cardiologist from Atrium Health. Thank you. Jack Parrish, owner of Sunflower Baking Company. Thank you. Scott McFarlane, senior staff engineer, Lending Tree. And the last one is, I don't have the name here, is another judge. This was a late addition, but we have a very important judge here, Tejashri Arote, Charlotte alum, software development engineer, most recently at Texas Instruments. Thank you. And thank you to the CGLL team, including graduate student Charmaine Hatimbai and Hannah Williams, and to all our graduate student volunteers today. Thank you. So as an urban research institute, we need our community to see more of the good work being done by our students. As we go to this R1 status, which is unbelievable, yeah, you can clap for that. We really appreciate all of you being here, and let us make this event really memorable. Thank you, everybody. And now I would like to introduce Dr. Jill Huerta, Assistant Dean and Director of the Reynolds Center for Graduate Life and Learning, who will kick off our competition. Thank you.
Thank you, Dean Mukherjee. Welcome, everyone. I'm so thrilled to see you here today. This is by far my favorite event that we do all year in the Center for Graduate Life and Learning for so many reasons. And every year, I kind of stand up here and I explain why it's my favorite event. And one of the reasons is because our graduate students who participate in this event really have to stretch themselves. I don't know if you've read some of the studies that I read that people might prefer to jump out of an airplane without a parachute than to stand on this stage and do public speaking. So to ask them not only to stand on this stage and do public speaking, but to talk about their research with a timer and to a general audience, to eliminate all the jargon that they normally use when presenting about their research. So I tell them, you've got to leave your comfort zone, you've got to stretch yourself, you've got to do bold things. And every year they do it, and I think that's remarkable. And 3 Minute Thesis wasn't around when I was in grad school. It's a relatively new competition. So I got to thinking this year that if I'm going to encourage these students to leave their comfort zone and to do things that are really hard for them, I should know a little bit about that. So this year, instead of giving you a relaxed speech about the value of 3MT, I'm going to do a 3MT spec. I can already tell you it's been an exercise in empathy because it's a lot harder than it looks from the outside. So as much as I appreciated you all before, I'm doubling down on that today. Because my heart is beating pretty fast right about now. Anyway, Dr. Kerr, if you could get the timer ready. And judges, no need to score. This is exhibition only. So, you already started. I didn't get the nod. Please stand by while we reset.
I'll just push it and it starts at 3 minutes.
Oh, got it. Good. Okay. Ready? There are few things in this world that are all kinds of good. But the 3-minute thesis competition developed out of natural disaster, namely a drought, in Queensland, Australia, is one of those. This competition spread quickly, and today, over 900 universities across 85 countries hold competitions like the one you're seeing today. Why? Why did 3-minute thesis spread so quickly? Because it's all kinds of good. It's good for our graduate students, learning to clearly and succinctly explain their research to a lay audience will prepare them for so many of the endeavors they're going to embark upon in their professional lives. Interviews with private firms, preparing effective proposals to fund their research, pitching their research when they have an innovative idea or product that they want to commercialize. 3MT helps them with all of that. Even more, this exercise of distilling their research to its essence and articulating its value actually deepens their understanding of their research, enhances their motivation, and drives them to do better work. And that's good. Secondly, 3-minute thesis is good for UNC Charlotte and good for higher ed in general. During this time, when higher ed is being asked more than ever before to justify its existence, it's critical that we have events like this one that showcase the magnificent, groundbreaking work we're doing here and the positive impact that has on our city, on our state, on our region, and beyond. That's also good. Finally, 3MT is good for society. We live in a time of complex and serious problems and challenges and opportunities. And the pathway to crafting pragmatic and effective solutions to those problems is communication. We absolutely have to have business leaders and scientists and policymakers and other experts able to sit down at the table and talk with each other and understand each other's work. 3-minute thesis was born out of natural disaster, but the skills it builds have the potential to save us from disaster. That is all kinds of good. And that's why I'm so passionate about this competition that I'm willing to stand on this stage and give it a crack with our students. Thank you. Okay. Moving on. So we'll get things kicked off here. As you know, Mukherjee explained the rules that there's no props, right? No notes, which is very typical, and one static slide. So students, please make sure that you don't bring anything up here with you. And the other thing to really pay attention to is the timer, because if you should run out of time, it's essential that you stop speaking right away, because otherwise you will be disqualified if you go over the buzzer. So with that, I'd like to invite our first presenter up to the stage. Our first presenter is Heather McBride, and she'll be presenting on expression of biofiltering genes across several species of endangered Uniondae. Competitors, when you're ready, you're going to give a nod over here, and we will advance your slide and begin the timer.
By a quick show of hands, how many of you have ever swam in a freshwater lake, perhaps in a freshwater river? All of you with your hands up, you have these organisms to thank for the quality of that water. I have had the pleasure of working to annotate the genomes of several species of endangered freshwater mussels over the past few months, and most of my work is computational, so instead of boring you with some code today, I'm going to tell you why my research is so important. Despite their small size, freshwater mussels are essential for aquatic environments. They are filter feeders, so they remove harmful things such as bacteria, algae, heavy metals, and other organic materials from freshwater environments. Without them, we've been seeing freshwater become too toxic for even some of the hardiest species to endure. Now, you all are probably thinking, why does this matter? Well, when you remove a keystone species, you're going to see the entire ecosystem struggle. We're seeing declines of their populations, and something needs to be done about it. Why are their populations declining? Issues such as pollution, habitat fragmentation, and competition with invasive species. The goal of my research is to look at these genomes and potentially compare them with invasive species that are thriving. We all know zebra mussels are one of the most thriving species out there. Why? What difference is there at the genomic level between the struggling native species and the thriving invasive species? With my research, if we can pinpoint that, hopefully we'd be able to target breeding programs or even habitat restoration efforts. So, while they may be small, they are a huge role and one of the strongest backbones of aquatic environments. And the overall goal would be to make sure that these species can be reintroduced and start thriving again. Thank you.
Next, we have Zina Golami presenting on application of AI in ophthalmology.
Imagine sitting at the eye doctor office. You are there for routine checkup. But what if I told you there's an AI machine that could detect earliest signs of vision-threatening disease long before you ever feel a symptom? Today, I'm going to show you how we are going to use cutting-edge AI with federated learning to revolutionize eye care, all while protecting your privacy. At the core of this innovation is optical coherence tomography, or OCT for short. OCT captures detailed images of your retina. It's like getting a 3D look inside your eye. These images are crucial in detecting disease like diabetic retinopathy, where early intervention can mean the difference between saving or losing your sight. But analyzing these images manually, it's time-consuming, error-prone, and overlooked. That's where AI comes in. AI models can analyze millions of these images faster with greater accuracy than human eye can. By early detection of disease like diabetic retinopathy, AI-empowered doctors can intervene, saving visions. But here's the catch. These models need large amounts of data to become truly powerful. And these valuable data are only available at hospitals. Hospitals are reluctant to share this information due to strict privacy laws. So, how can we have the benefit of both worlds, access to these valuable data without compromising sharing concerns? Federated learning, a game-changing solution. Federated learning allows multiple hospitals to train the AI model collaboratively. So each hospital train the AI model using its own local data and only share the model updates. It's like sharing a recipe without sharing its secret ingredients. But federated learning also comes with its own challenges that we are addressing in our research. The first one is the trade-off between personalization, how should my model behave in a particular setting, versus generalization, how should my model behave in every setting for everyone. The second one is the aggregation strategy. How should we aggregate these model updates coming from different hospitals to get the best model possible? In the end, the future of ophthalmology will be shaped by AI and federated learning. Together, we can create an eye care system that is faster, accurate, and accessible. Thank you.
We have Dylan Moretz, who will be presenting on unlocking biological insights, visualizing big data with the integrated genome browser.
Have you ever tried solving a puzzle with missing pieces? Frustrating, right? This is the challenge researchers face every day when working with genome data. Now I know what you're wondering. What is a genome, and why is it so important? To put it simply, a genome is an organism's entire set of genetic instructions. It determines why some people have black hair while others have brown, why some wear glasses while others don't, why some are tall while others, like myself, are quite short. It explains many of the things we observe with the naked eye, but it also reveals the things that we can't see, like why two individuals prescribed the same drug have different reactions, or why a crop will perform better than another under the same weather conditions. Interpreting and analyzing genome data is crucial for advancements in research areas such as medicine, agriculture, and evolutionary biology. However, current tools are struggling to keep up with researcher needs. For example, the genome data inspection tool I worked on during my internship, the Integrated Genome Browser, struggles with detecting genome assembly issues. These issues can result in misleading or inaccurate findings. To combat this issue, my team reached out to users of the software to determine what their challenges were and what features needed to be added. We then coded and designed these features and integrated them into the existing software. And finally, we tested it with data sets provided from research teams around the world. And the results, a more user-friendly, streamlined user interface, better data accuracy, and tools that empower researchers to find more reliable insights from their respective data. Much like a puzzle, solving complex genome-related research questions requires having all the right pieces. With the new updates that we've made, researchers are now more equipped to solve their respective puzzles. Thank you.
Sarah Tazar, peer-led sexual health education for older adults in congregate living.
Good evening. Approximately 10% of individuals in their 90s are sexually active, and that percentage increases as we roll back the decades. Additionally, as you can see here, the rates of sexually transmitted infections in older adults are rising. Additionally, by 2025, 20% of individuals in North Carolina will be over the age of 65. Despite all of this, sexual health education for older adults is tough to find. We cannot simply take sex ed from middle school and apply it here. Older adults aren't worried about pregnancy, and suggesting abstinence as a form of infection prevention is patronizing. It's time for a targeted intervention. Peer-led health education increases community buy-in as well as increasing positive health outcomes. As such, it is the basis for this research project. I've recruited a community action board of 5 older adults who live here in Charlotte, a public health professional from the Department of Health and Human Services, a community educator who specializes in working with older adults, as well as the director of programming at the community that we're in. Together, we've created a program to talk to older adults about sexual health that is relevant to their age and stage. This 2-session program is called Safety and Satisfaction. Session 1 starts with the STD-KQ, a questionnaire to establish a baseline of STD knowledge. Then, the 2 peer leaders who are from the group that helped develop the program will lead participants through conversations about the benefits of staying sexually active, the most common sexually transmitted diseases and their symptoms, the physiological changes that occur as we age and the impact on infection transmission, types of protection that are available, as well as where and when to get tested for STDs. Session 2 covers sexual ageism, which is the stereotype that older adults are non-sexual, what its impact on our health can be, as well as how to combat it. Then, the participants will be separated into small groups and given different scenarios to role play in order to practice having these conversations in an environment where they can get feedback. Finally, the participants will be given the STD-KQ again as a post-test to show that this program works. I will then analyze that data and use the results to make improvements and as evidence to spread this program across North Carolina. Thank you.
Our next presenter is Brandon Bendixson, and he'll be talking with us about mitigating bias in the genomic analysis of several tomato varieties.
Hello everyone. Tonight, we're talking tomatoes. Did you know that tomatoes are the second most consumed vegetable in the United States, second only to potatoes? What this statistic shows us is that we Americans love our tomatoes. However, the rise of global temperatures is threatening our beloved tomatoes. Elevated temperatures can impair plant performance and reduce crop production. On top of that, tomatoes grow smaller and deform when exposed to acute heat stress. However, some varieties of tomato are more thermotolerant than others, and that is where my research comes in. I'm working as part of the Tomato Genome Project, which is a collaborative initiative aimed at identifying genes that make some tomatoes more thermotolerant than others. My research focuses on reducing bias in the genetic analysis of 4 tomato varieties. One of those varieties is Heinz, which you probably could guess comes from the Heinz company. While this variety makes great ketchup, it is much more sensitive to the heat than the other 3 varieties. So, in the original experiment, all 4 varieties were compared to a Heinz reference, but identified genetic differences. While this method did provide insights as to why these 3 varieties are more thermotolerant than Heinz, it introduced a major problem. You see, there might be genes or genetic differences in these 3 varieties that simply don't exist in Heinz. So, by using Heinz as our only reference, we run the risk of missing those unique genes. When we miss those unique genes, this causes an overrepresentation of genes similar to Heinz across all 4 varieties, which means our results are biased. To address this problem, I generated 4 unique references for each variety and compared them to themselves. While this method did help, it still did not fully eliminate the bias. So, we shifted our project and are now going to compare our 4 tomatoes to a totally new set of unrelated tomatoes. This should allow us to identify new genes and further minimize bias. But this shift in our project has an important lesson. Research demands flexibility. By reevaluating our strategy, we have been able to advance our project and are moving closer towards meaningful results. All that said, let's take a step back and look at the big picture. Global warming is threatening food security by reducing crop yields. The impact of this work is that breeders should use our research to develop tomatoes that are resilient to rising temperatures, which helps sustain productivity in the face of climate change. Also, the insights gained from my research have the potential to extend well beyond tomatoes, as our research offers valuable lessons for improving other crops as well. We live in an era where sustainability and agricultural innovation are critical, proving the Tomato Genome Project is just one part of a much larger global effort. Thank you.
Next, we have Lauren Holtquist presenting on Keyboard Warriors.
Everyone here uses the internet. And that means that everyone here is at risk for harassment, stalking, and the leaking of our personal information online. Everyone here also has a personality. And there are some personality traits that seem to be related to higher levels of aggressive behavior online, like narcissism and spitefulness. There's also research that says that when people are anonymous, it makes it a little bit easier for them to do terrible things to other people. So we put all of this together, and we decided to research how personality and anonymity impact aggressive behavior online. What we did was we gave people, first off, a personality test. We asked people how they tend to behave in their daily lives. And then we sorted people into 2 different groups. If they were anonymous, they didn't share any information about themselves. And if they were identified, they had to give a selfie and a short personal description. And how do we measure people's aggressive behavior? Well, we put them in a button pressing competition. And there were 8 rounds, and when they won a round, they got to noise blast their opponent as hard as they wanted. The more loud the noise, the more aggressive. So what we found from this was that when people are anonymous, they're more aggressive. And that when people are higher in different dark personality traits like narcissism, they're more aggressive. But we also wanted to look at the combination of the 2. And what we've found so far is that when people are higher in a dark personality trait like narcissism, and they're anonymous, they're even more aggressive. The only personality trait that this trend did not hold for was spitefulness. Spiteful people don't seem to care if they're anonymous or not. All right, so what does this mean for us? Well, we found out more about how anonymity and personality impact aggressive behavior online, which is great because this is the first study that's actually looked at people's behavior instead of just asking them survey questions. And we're going to be doing some more research about how people's motivations are different for aggressive behavior online as well. Of course, this leaves us in the current day. And we aren't going to get rid of the internet. We can't get rid of personality. And we also shouldn't get rid of anonymity. Because anonymity also protects us. The first thing people do online when they want to harm others is find out all of their personal information. Their name, their home address, right? And so in a somewhat ironic twist, the best practice that we have as individuals to protect ourselves online is to stay anonymous. Thank you.
Our next presenter is Vedika Giudiani, and she will be presenting on AI-driven insights into viral recombination.
Imagine a virus swapping parts of genetic code, like exchanging pieces of puzzles. This process is called viral genome recombination. This is one of the main ways through which viruses evolve, adapt into new environment. Sometimes it makes its new version of itself, and in some cases, it can even lead to pandemic. My research is using artificial intelligence to detect these viral recombination patterns quickly and accurately. But the question is, why is it even important? Well, understanding these viral recombination patterns will help us to track the history, to see where and when these viruses has changed their behavior, how they are causing these deadly diseases, and it is also helping us to better prepare for any future viral outbreak like COVID-19. To better understand, let's see an illustration on left, where one virus donating its piece of puzzle to another virus, making a recombinant. This recombinant can behave differently than other two viruses because this has got mixed information from both the viruses. This recombinant can have different symptoms than other two viruses. But how do we see that? Well, the way we have our family history, viruses has their own. So we can make family tree for viruses by the process named phylogenomic analysis, as you can see in the middle. Through this process, we can see how one strain of virus is connected to other strain of viruses through their genetic code. And it will help us to see exactly when those viruses started changing their behavior. But this process is very slow and complex. That's where AI or machine learning comes into the picture, as you can see in the right. We are developing a machine learning model which is going to analyze this data, and it is going to collect all these characters together on the basis of their similarity. And by automating this process, we can track massive amount of data very quickly. In short, we can say my research is seeing how viruses are changing, and it is preparing us for future viral outbreaks. Thank you.
Our next competitor is Caroline West, and she'll be explaining to us that there's a virus in my bacteria, in the fungus, in the plant.
Who has a favorite virus? Just a few of you? Of course, how could you possibly choose just one? Well, I might be a little bit biased, but my favorite virus is one I found in a bacteria, in a fungus, in a plant. Now this begs the question, why do we care about some weird virus in this convoluted system? And the reason why is this fungal host, Rhizopus. Rhizopus is a known plant pathogen, but it can also cause disease in humans who are immunocompromised. such as cancer patients, people who've had transplants, and the mortality rate is 70 to 100%. The concerning part is we don't fully understand how this Mycelia habitans and rhizopus system causes disease. And that's where the virus comes in. We've known for years that many deadly bacteria infections are actually dangerous because they're viral DNA. One example is cholera. So cholera is a deadly bacterial infection that leads to vomiting, diarrhea, but the cholera toxin itself is actually from a virus. And so if you get cholera and you get the bacteria with the virus, you're going to be much sicker than if you get the bacteria without the virus. So that made me wonder, is there possibly a virus leading to these symptoms in the rhizopus infections? To test this theory, I ran 20 different strains of Mycelia habitans through a computer program. I looked for viral DNA, and every single one of the strains had some viral DNA, with over half of the strains having enough viral DNA to make fully functional viruses. So next, I went to the lab, and I wanted to test these computational predictions. I took 2 strains of my Mycelia habitans, and I stressed them out. That's what you do when you're trying to get a bacteria to make its viruses. You stress them out. So you want to stress them out just enough so they make their viruses, but not too much that can kill the bacteria. I found that UV light worked the best. And after 48 hours of exposing my bacteria to UV light, we saw what looked like to be viruses. Now, we do need to run more tests to make sure these are actually viruses, what these viruses look like, and what these viruses do. But the preliminary results are really promising. If we can figure out that this virus is responsible for the toxic effects of Mycelia habitans rhizopus infections, we can start to make drugs to target this and lower that mortality rate down from 70%. So next time you're at a party and someone asks you the inevitable question, hey, what's your favorite virus? And you go, oh, it's that virus that you find in a bacteria, in a fungus that infects plants and sometimes humans. Thank you.
Next, we have Emanuela Lixindai Izquierdo, presenting on identity negotiation for parents in multilingual families.
My native language is Albanian. My husband is a mathematician from Colombia. We never would have thought that these were the first things we wanted someone to know about us until I got pregnant. Being a linguistics student, I was aware of the power of language, but I had previously never felt such a strong emotional connection to my native language. Surprisingly, that was not the case for my husband. And it made me wonder, how do other families like ours, families in which the parents speak a different language from one another, and live in a country where a third community language is spoken, how do they talk to their children? And what do these decisions tell us about what parents value, what aspects of their identity express, and what do they imagine for their children? The number of these families has grown exponentially in the recent decades as a direct result to the unprecedented rates of growth of globalization. And yet, the research on the language practices of such families is lacking. And so, to answer my questions, and in the hope of contributing to the field, I decided to conduct interviews with said parents. And I turned to social media, which is actually an accelerator of globalization, and I was able to get in touch with parents from all over the world who, just like me, had grappled with these questions and, unable to find answers, had followed their heart. I conducted the interviews via Zoom, and now I'm in the process of analyzing them to see what ideas are shared among all interviewees. Turns out that there's a very strong correlation between an individual's love and aptitude for language and their tendency to seek a partner that speaks a different language, and then a desire to teach languages to their children. And if the parents' initial goal was just language, they had started to incorporate other cultural elements like food and attending festivals. Another common theme was parents reporting this initial fear of confusing their children with too many pieces of identity puzzles, which had turned into a belief that their children did not need to belong to one or two boxes. Rather, they could be citizens of the world, So to everyone concerned that globalization is killing individual cultures and to parents fearing about confusing their children, I tell you that multilingual families are a beneficial product of globalization and an advocate for a more connected world where differences are seen in order to be celebrated. Thank you.
Last but not least, I'd like to welcome Maitri Patel to the stage to present on bacterial triglycerides increase agricultural productivity, a computational approach to improve microbial viability.
Farmers use chemical fertilizers to improve plant performance. These chemicals are not only harmful to us, but they're harmful to the birds and the cattle that feed on them. They also seep into the soil and pollute the water table. So, how do we make agriculture more productive, yet safe and sustainable? Well, the answer to that question are the trillions and trillions of microbes present all around you. But when microbes are sprayed onto the fields as fertilizers, they experience harsh weather conditions. For example, if it's drought-like and if it's really hot, they can dehydrate and die. So how do we improve not just plant productivity, but also microbiology? This was a research question that my team and I sought to answer during my internship with a company called Three Minute Thesis. Towards this, I developed a bioinformatics workflow. So I first took the genome of 5 bacterial species. These genomes, they consist of thousands of genes. When these genes are turned on, they produce proteins. The proteins are involved in pathways that perform specific functions for the cell. So after I got the genome, I asked the question, are the genes that are linked to viability present in them? If they are, what pathways are they present in, and are these pathways present in completeness? If the answer is yes, I define a growth media that I think the bacteria will do well in, and I computationally simulate stress conditions. And then I see, does the microbe do well? Does it survive longer? What I found in one bacterial species was an energy storing mechanism. Like how in us humans, we produce and we store fats, and then we use those fats later on for energy production. Similarly, this bacterial species, when it was grown in the media that I described through this workflow, it turned on the genes to produce bioplastics, which were stored in the cells as energy reservoirs. This could be used by the bacteria in the field under some stress. So using this workflow, you can screen for genes and pathways that are important for the microbe. This can reduce time spent in the lab that you perform doing the same experiment that basically tell you the same thing, but they could be a hit or a miss. They also take from months to up to a year to perform. So this workflow result could confidently test results in the lab and then into the field. So if you were to implement this workflow to the numerous microbes that are beneficial to the plants, then one gene at a time, we can make agriculture more productive, yet safe and sustainable for everyone. Thank you.